Welcome to Jason Live. I'm Haley Nelson, and we are back with our STEM career series, where we learn about careers in science, technology, engineering, and math from role models in those fields. And today's role model is Dr. Eric Benbow. Now, he studies microbes on dead things. He teaches, he researches, he does field work, and we're going to learn all about our STEM role model and more when we connect with Eric in just a moment. But first, I want to remind you that today's event is live and interactive. So if you're joining us from the Jason Learning website, you can see a little box right under this video window where you can submit your questions, your comments, or you can tweet us at #JasonLive. All right, now it's time to meet Dr. Benbow. Hi, Eric. Welcome.
Good afternoon. Thanks for having me.
I'm so glad that we could be here asking you these questions today. And so let's just jump right in. Would you tell the students a little bit about what an ecologist is and an entomologist in general?
So an ecologist is basically anyone that studies interactions in nature, whether that's birds or reptiles or fish, or in this case that I study, insects and microbes. And insects are basically an arthropod invertebrate, right? Doesn't have a backbone, has an exoskeleton, 6 legs, and many appendages. Anything from flies to a grasshopper to, I'm at a loss, right? Creepy crawlers. And most people call them bugs, right? But bugs is only one specific type of insect that we typically call hemipterans. But yeah, so we study insects, and then we study how microbes, microscopic organisms, those organisms that you can't see with the naked eye, but you have to have assistance with the microscope. We know that they play a major role both in human health, right? How you get sick, how you function, right? But also we think that they have a tremendous impact on how insects and other creepy crawlies function as well.
Well, Sophia Flores from Congressional wants to know, and you were touching on this a little bit, but how does your job affect the world?
So a long time ago, I was asked, so what, who cares about that bug, right? And I didn't know how to answer it. I know about a lot better now. Studies on insects and microbes themselves affect almost every aspect of life, whether it is West Nile virus or malaria, and how insects spread those pathogens, termites and the cost of termites, sometimes in forensics. So we can use insects in death investigations, determine maybe how long someone's been dead, right? And that has tremendous importance. Medicine, there are wound debridement. So they actually place larval maggots, fly maggots onto a wound. They don't hurt the skin, but they eat the bacteria that can cause infection, and they eat away the dead tissue in those particular cases. Fishing, huge industry in fishing, especially here in Michigan. Fly fishing, you understand when certain insects emerge from aquatic habitats, because when they die, they fall back onto the water. And fishermen, fisherwomen, right, fisher persons, tie those to represent the insects in nature so that they can catch the big fish. Multi-million-dollar industry in the United States. So anything from health to recreation to industry, and maybe how insects can be used as alternative food sources for aquaculture and livestock in different parts of the world. So studying insects can have a huge impact on a variety of aspects of here in the US and globally.
Touches on so many things. Mrs. Flynn says, if bacteria are able to evolve so relatively quickly in general, how is it that the microflora that live in and on humans have not evolved somehow to destroy us?
That's an interesting question. In some cases, they actually have, right? Many strains of infectious bacteria are becoming resistant to the antibiotics that we've produced over the last 150 years. Those that do resist all forms of antibiotics can and often do kill people. Maybe not often, but they are killing people. So right now it is an arms race. With the evolution of both humans and bacteria, we have co-evolved, right? Our immune system is constantly adapting and evolving to new pathogens. That's why it's so important to get vaccinated for new viral pathogens every year, such as the flu virus, but also for some of these strains of bacteria.
Wow. Well, we've got Sophia, and she says, what school subjects do you apply most in your work?
Man, entomology.
I bet she's thinking about, she's in middle school.
Absolutely. No, actually, biology, chemistry, and math.
All right. Students, we want to remind you that during the live broadcast, you can ask whatever you like. We've got lots of really, really cool things that Dr. Benbow does. So let's see, we have what inspired you to pursue your career?
When I was probably in middle school, I would spend my summers in creeks in Ohio, and I would be trying to catch crayfish and little minnows. I got pretty good at it sometimes. And I enjoyed being outside. When I was in college, I wanted to be a doctor, a medical doctor. And I was taking chemistry, biology, physics, all those things that were necessary to get into medical school. However, I took 2 courses where the professor took us outside. We went on boats on lakes. We went into forests and identified the plants and the insects as well. And I knew right then that's the kind of job I wanted to do. And so I essentially pursued that now.
So the field study really is what hooked you, getting out there, collecting data. You like that aspect of it. And how do you balance that with what you have to do in the lab?
So the research that we do in my lab is very well balanced between field work and laboratory work. Some of the necessary field work is probably the more enjoyable part for many of us, because we get to see new and exciting places. But we bring those samples back, and now we're bringing those samples back for molecular analysis of the microbes, and the bacteria, the archaea, the microeukaryotes. That is much more stringent and scripted type of research where you follow direct protocols. In the field, you have to make decisions with changing weather and changing environments. In the laboratory, protocol is what essentially designates what you're doing.
We have a question from Mrs. DeMayo's 2nd grade class at Salem School. Are there insects that are helpful to people?
Yes. Think about honeybees and other pollinators. They pollinate and essentially become functionally important to huge amounts of food crops. So the food that you eat every day, most of that, especially vegetables and fruits, are pollinated by insects. That's one major example. Mosquitoes that transmit disease and are annoying, right? Incredibly important to human health. Malaria used to be endemic throughout the United States. It's no longer there because we've eliminated many of the mosquito populations that actually had the malaria parasite.
What good do mosquitoes do?
They act as food resources for other insects and bats and birds in the environment. As larvae in vernal pools and in container habitats, they actually function to reduce bacteria and change water quality as well.
What would a student think, this is a question from another guest, what would a student think is the coolest thing that you do? Possibly the most disgusting thing.
We will we will sometimes collect insects from dead people.
So, I mean, and how do you go about doing that? What's the process that you do? Do you just, is it people who have donated themselves to science, or is it tricky?
It is tricky. It's done in a variety of ways. Some are with bodies that have been donated, where the person has designated that their body be used for scientific research of this sort. And there's 4 or 5 places in the United States that allow that, and we've done research in those areas. And so you collect the insects as part of research experiments. Another part of that is forensic entomology directly applied. So meaning we can be called to cases, we have gone to cases, where you go to a crime scene or you go to a death scene and you approach, after being cleared by the officers, you approach the body and the remains and you collect the insect evidence from the body or from the soil or habitat around or near the body. And then we use those insects, we identify the species of the insect, we look at how big they are in their life stage, and then we take the temperature or understand the temperature of the scene, and we can use that to model how long someone's been colonized by those insects.
That is so incredibly interesting. It's like you're living on an episode of Bones.
Yeah, Bones and CSI. I know a couple of the consultants for those shows, actually.
You could be one of the consultants for that show.
Yeah, could be.
You could base one around something that you've studied. That'd be great. We have a couple of students, Aaron and Alexa from Oceanside Middle, say, what do you think is the most interesting type of insect you have studied and why?
Most interesting type of insect. So there's there's a whole bunch of them actually. But perhaps the most interesting is the insect that I studied in Hawaii that inhabits the lips of waterfalls and cascades. It's on there right now. So that inhabits these waterfalls, and you see the white material coming out of its mouth. They secrete this sticky substance. It's like silk, but it doesn't break down in water, and it allows them to adhere to rock surfaces with high velocity water. I just saw today actually on Facebook that another type of insect that does something similar, that chemical engineers are researching that particular compound, the silk that's coming out of their mouth, because they also use it in aquatic habitats. What would be really cool is if you understood that silk, much like spider silk, right? But you could use it for maybe wound covering and wound treatment as well. So that's one of the insects that I think's most interesting to me.
Wow, and there are a bunch of entrepreneurial endeavors that could use technology like that as well.
Absolutely.
We have 2 questions. Let's see, this one is from Dark Rider. What's the hardest thing you've ever done in the field, and what's the most difficult thing you've ever done?
Most difficult thing in the field has been in West Africa when I forgot to wear a hat, and we were sampling for insects, aquatic insects, and a pathogen, a Bruley ulcer. It was 110 degrees, no clouds. I didn't bring enough water, and I almost passed out. That was probably the hardest thing I've done in the field. One of the job-related most difficult things that I've ever done was sifting through human remains that had been found in a slow-moving, low-oxygen, warm river here in Michigan. The smell is unforgettable, and it was very profound. It's difficult.
Just out of my curiosity, have you ever had to use the beetles that take all the stuff off of bones? And I mean, we see that on TV all the time. What is that about?
We haven't used them here. I have an anthropologist colleague at Michigan State that we've talked about doing it, but they're commonly used in museums to essentially consume the tissue, soft tissue from skeletal or skeletonized remains to get down to the bones. But I've never done it myself.
We have another question about bugs that you do or do not like. Is there an insect that you do not like at all?
Not really. There's some spiders, arachnids, that I do not like. But insects, Yeah, insects. There's some
Or
crustaceans.
maybe one that you're like afraid to be near. I guess that would be.
Um,
I don't know.
Mildly allergic to ants, and fire ants has caused me to go into anaphylactic shock. So, yeah, ants I stay away from.
Oh, so you work where you're dealing with different insects that are, passing around pathogens. Do you ever worry that you are going to get one of these diseases or be infected by what you're working with?
Absolutely. Working in West Africa, there's malaria. There's all kinds of viral pathogens that are transmitted by mosquitoes. And the insects that we were specifically studying that transmit a microbacterial pathogen that causes major ulcers on of the skin and specifically of children. Both my colleague and I were bitten by those insects in areas where the disease was very prevalent. And that they bit me through some gloves that I had on as well. And so, yeah, I think about that a lot, especially in those areas of the world where there's lots of infectious diseases. In that particular case as well, we were in water that we knew was positive for schistosomiasis, which all that takes is putting your arm into the water. Nothing has to bite you, but the little cercaria embed into your skin. And so that is, it can be disturbing sometimes thinking about where you're at. Nonetheless, the crocodiles that are 25 meters away in the same habitat as you.
Well, how do you manage the prevention of that? Because if you've gotten bitten, were you just lucky and the one that bit you wasn't infected, or do you prevent somehow? I mean, if it can bite you through your glove.
Yeah, they don't. These particular insects don't normally bite you. They're not aggressive. But I was picking up plant material, and it felt alarmed, and it was defending itself. It didn't have the pathogen, so in that sense, I got lucky. But also, part of what we were studying was to determine whether or not those insects actually could transmit the pathogen. There were some laboratory studies, some anecdotal evidence, and we were trying to demonstrate whether or not they actually could. And that still hasn't been determined. I think I just got lucky.
I'm glad you're lucky. We have a couple of questions coming in. Do you travel a lot for your work? What are your favorite places to go? Are there any that are your least favorite? And why do you go to other places to study what you study?
Oh, that's a good question. I do travel a lot. I used to travel a lot more. And West Africa was a place where we had to go to study this particular disease. The disease also was found in Australia, so we were able to travel to Melbourne, Australia to study it there as well. The research that I've done in Hawaii, that was part of my PhD work, and we had to go to, there's only certain organisms are found in Hawaii, and they were being affected by water flow, so we went there to study those organisms. The forensic entomology and the microbial forensics that we've been doing has been taking me all over the world as well, from Thailand to China to Europe, and actually now some parts in Africa and South America. Much to my wife's chagrin, sometimes I've been traveling too much for some of those things. One of my favorite places would probably be Thailand. For a variety of reasons, sociocultural reasons, but and it was one of the places I had never been to frequently before I went.
Do you get to take your family with you ever? I know you have 2 daughters. Is that their names on the board back there?
They have not gone yet, but my oldest is very much interested in going to Africa. And we've been figuring out when the best time that would be. But here in the US, they come with me almost to every place I go to do research, whether it's a decomposing pig or, as you can see there, in the bug house here at Michigan State University, where they get to interact with insects, both in the laboratory and at home and in the field. So yeah, here in the US, they go with me a lot of places.
So in general, you're collecting a lot of data from a lot of different kinds of animals and humans. Are you taking a kind of a general survey or different parts of different animals, and how do you keep track of all of that data and be able to have kind of a stash of things that are similar, things that are different? And are you tracking them across the world or in one certain area? I've got a million questions.
Yeah, no. So the central question that we are asking is, how do microscopic organisms affect insects? And how do insects affect the microscopic organisms? And we've really centered that around decomposing things, whether that's a human body or a pig or leaf litter, right? Leaf litter in a stream is the energy source for that ecosystem. And the microbes and insects interact to break down that energy source, those leaves. And that affects the quality of the watershed downstream of it. It's a huge amount of data, and we take incredible care to be highly organized in, one, how you collect the data from the field, how it's processed in the laboratory, and then where those data are archived on massive computers, computer systems, and networks. And then ultimately, how you analyze the data requires a lot of quantitative expertise that we have in the lab. But yeah, it's difficult managing. But if you think about forensics and decomposing organisms versus microbes and vectors, such as the diseases in Africa, right? Or just carrion flies and microbes. And that's a whole other aspect, but it's essentially related to things that are decomposing.
We have a question that says, can a velvet ant actually kill a cow?
I'm not sure.
Well, that's what we're going to have to look it up. Somebody look it up.
That's a good question.
Yeah, there we go. And somebody says, what are some good ways to identify whether an insect is poisonous or dangerous?
You can't always tell, but often color, bright colors, extravagant appendages. So for instance, in caterpillars, they'll often be brightly colored, or they'll have the little setae that come out that have poison on them, right, and irritants. And so in general, but not always, it's the color and the different types of parts on the body that stick out so you see it, right? Because the reason they want you to see them is because they don't want to be bothered. And if you bother them, they will sting you or otherwise harm you.
That makes sense. Well, do you have to essentially be on call for going out to find these decaying dead things, in your certain area? Or if you go somewhere else, if you're in Africa, are you just waiting for things to die to go do your research? I don't know how to say it exactly.
Yeah, in some ways, we did a forensic case a couple of years ago. And essentially it was, someone called me up. I was sitting here, not doing an interview like this, but sitting at this computer, and they said, we found a body and there's insects all over it. We need you to come down if you can and collect those insects because we think they might be important for the evidence. And so we did. Jumped in the car, went down there, essentially in this outfit, essentially the same type of outfit, and collected the evidence. Right now we're on call with the Flint, Michigan's medical examiner's office for similar cases. And sometimes we get evidence sent from other parts of the world and other parts of the nation where they want us to evaluate the insect evidence for determining a postmortem interval or something similar.
So when you go, and I guess I have a question in general about, is there sort of a standardized colonization process after someone or something dies? Like you see this first, and then you see this microbe, etc.
Yeah, there is.
How you can tell what the timing of their demise?
Yeah, so when something dies, right, whether it's a human or another animal, within minutes, flies can show up. Just house flies, normal house flies, are also called blowflies. They'll be there very quickly. We've had dead organisms have the insects there within 15 minutes. Some people have reported within 5 minutes. So if something's flying around your head, no, I'm just kidding. But the flies are the first to get there. They lay their eggs on that resource, and those eggs hatch into maggots or larvae. And those larvae grow at a rate that's based on temperature. So the hotter the temperatures, the faster they're going to grow. And they go through several life stages. And at the end of the third part of the life stage, they start migrating from the body or the resource. And they try to find a dark place to get away from predators. And so, the blowflies are the early colonizers, right? It means recent death. And then beetles start to come in. Beetles, wasps, they come in to either breed on the site and the resource, or they come in to eat the maggots that are there, right? And then essentially they become an indicator of later parts of decomposition or longer time since the individual died. And then you get another set of beetles that's later, that's just related to the skeleton and maybe hair and skin, dried skin, mummified parts. And those are indicative of really longer periods of time. But if you go back to the flies, right, and how they're developing on the carcass, you can get a certain size of one of those flies, and if you know the temperature, you can determine how long they've been dead. If they weren't concealed, the body wasn't concealed. Flies will get there within 15 minutes to maybe an hour or maybe 3 hours, right? There's some variability there. They'll lay their eggs, and that's the colonization. But if someone has killed another person, has somehow concealed the remains, or let's say wrapped them in plastic and put them in a freezer, right? Now, they may wait 2 to 3 months, 2 years down the line. Now let's say, put the body outside. Flies immediately colonize. Now it's not the amount of time the person's been dead, right? It's the amount of time they've been colonized by the fly, which in this case would be a 2-year difference. They were killed 2 years ago, frozen, and then 2 years later, they were exposed to the environment, and then the flies colonize. It's a very important difference in a courtroom.
Wow, and so you've testified for I guess events kind of like that. That's kind of incredible. As someone who studied ecology and entomology, did you think you would end up here doing what you do?
No, not at all. I started studying insects in Hawaii and how water flow affected them. But what part of that study was interesting was I understood the life cycle, how quickly they grew in different environmental circumstances. Blowflies on a body, it's their life cycle, and it's understanding how the environment affects how fast that organism grows. And in Hawaii, it was waterfalls and biofilms on waterfalls. With forensics, it's a decomposing carcass. That's the resource, and we're evaluating how insects develop on it. The commonality is both have been useful to humanity. And in Hawaii, it was water resource issues. With forensics, it's determining if someone's lying or not, based on alibis.
Well, we have a couple of more questions here. Let me just kind of go back up here. Sorry. Let's see. What is the largest, this is a question from a second grade class, what is the largest insect you have ever seen or worked with?
For me, it's called a bellostomatid, or toe biter. And they get to be about that big. And they have a proboscis and a stylet, and they pierce a fish, they pierce other insects. There's rumors that someone saw, because they also fly, someone saw one about this big flying, and it took a bird out of air. and pierced it, and they sucked the fluids out of the organism that they're preying upon. It's something out of a horror movie, actually.
I'm terrified, actually.
I wish I had one, but yeah, about the size of a lighter. And those are found all over the world.
Wow. Oh, we have some information about the cow killer ant.
Okay.
It says the pain is so much it could kill a cow, but someone made a velvet ant sting them and didn't even faint, though he was super in pain by the look on his face.
Of course.
Miss Becky asks, how would you describe the microbiology of a healthy human before they're dead and decomposing?
Oh, there's some tremendous information out there right now. And in fact, we've been inspired by the Human Microbiome Project to do some of the work that we're doing now on dead people's microbiome. But what we know from the living human microbiome, the gut microbes in your belly, right, in your intestines, they affect anything from your overall health, obesity, cancer, certain types of cancer. It influences cancer and how you respond to it. Your cravings, behavior in many cases, how one responds to stress. We know as you grow, your microbiome changes. We know in infections, and when you are given antibiotics, it changes your microbiome, and that has changes onto your immune system. It's pretty profound. I think personalized medicine is going to be transformed by understanding the human microbiome, the healthy human microbiome, but also the microbiomes of those with ailments and issues. Some studies have suggested that even mental disorders, some mental disorders may be influenced by your microbiome as well.
So we've got microbes. We've got microbes living all over our bodies, living inside our bodies, some being helpful, some being not so helpful. And you're studying people and animals all over the world. Are there microbes that would be found on my face that would not be found on somebody else's face who's from a different place? Or even like culturally, I've read a study about microbes being found on people of different races being different, and I'm just wondering about those kind of things, because I think that's incredibly interesting that we're our own ecosystem, housing and different places on the body too, like under your arms versus in your ear, that kind of thing.
No, absolutely. And there's been some really good studies showing cultural differences of microbes, usually the gut microbes. That's what's been primarily studied. There's been a few studies that you've just mentioned that have shown the variability, how the microbes on your forearm is very different from your eyes versus other parts of your body. That is at the cutting edge of research in many ways, is trying to understand how your microbiome is unique to you, unique to the people that you live with and around, and perhaps how it changes when you travel. We actually have some studies going on with that. And in fact, how your skin microbiome may affect how insect vectors are attracted to you or not attracted to you, which is interesting. A couple of new studies have actually shown, and this actually gets back to forensics as well, you're shedding your microbes. As you sit in that chair, and all the kids around the country that are sitting in their chairs, they're shedding microbes around them. And there have been studies that have shown you go in and you swab next to a chair, you can identify where those microbes came from. Like, really good probability that the microbes that you just found in this room came from the person that was just sitting in that room for 3 hours.
So if you can't necessarily find DNA evidence, but you found their microbes, that could be something that you'd really use as forensic evidence possibly in the future?
In the future. And they're also showing the keyboard that you use, you shed microbes onto the keyboard and they can trace it back to you. Your cell phone, they can trace a cell phone back to you. Now there's time limits.
My mind's being blown. This is amazing.
I've been learning more about it because we are in a partnership with the Wayne County Medical Examiner's Office. So that's the office where when someone dies, they do autopsies. We're partnering with them because their office is swabbing bodies as they come in during autopsy. And we're doing what you said. We're swabbing the eyes, the nose, the mouth, the ears, a couple of other places on the body. And we're finding that we can use the microbes to determine how long someone's been dead in rough time frames. But we also think maybe very important lifestyle characteristics. If you're at higher risk of homicide, do you harbor a different microbiome because of a lifestyle that you're part of, right? Not necessarily linked to all the time, but you're a part of. And it's showing some real promise. Other studies have shown that the microbiome on your shoes can be traced back to where you've been as well, which is for a certain amount of time, which is pretty interesting. So there's tremendous potential of the human microbiome for health applications, but we're also starting to see the potential, it's not used yet, in forensic investigations.
So that question at the beginning, you said some people ask you, who cares?
Yeah, exactly.
There are so many different fields that this could absolutely revolutionize, but we have to do what you're doing and collect the data and find out, I guess, what the baseline is for all of these different areas, approaches. This is amazing. Anyway, I've done my 2 amazings, and I'm going to not say it anymore. We have a question. Was that a picture of you climbing a waterfall? Are you collecting data, or are you just having fun?
That particular picture, I was collecting data for my PhD.
Sure you were.
Well, I did swim across that pool, and then I climbed up on that rope, and I probably didn't need to sample from there, but it was a pretty fun day.
Awesome. We have a question from Mustangs. Why don't all bugs like dirty or decaying environments? What are the decay-seeking bugs looking for?
So why not all bugs? Because only a certain number of insects have evolved to be attracted to the odors and those types of resources, those decaying resources. Others have been attracted or evolved to be attracted to your breath, right, with mosquitoes. They're in your heat signature. Others have been attracted to flowers and the scent flowers give off, right? And others that have evolved to be in streams and rivers, lakes, etc. The ones that with decomposing organic matter, we're finding, we're getting more confident that they're actually cued to the bacteria and the microbes of that decomposing resource. And so what they're getting there is they're eating the tissue and the energy and nutrients that are in that tissue, but they're also eating the microbes that are there. And we think that the microbes are a signal that the resource or the tissue is available. And then they've evolved to use that to reproduce on and to develop offspring on. And so it's a natural evolution of an insect group that specializes in decomposing things. Now imagine if we didn't have these decomposing insects and invertebrates. What would happen when you have say a salmon run and tens of thousands of them die? They get flushed to the ocean, some crustaceans eat them. But what about all of the cows that die, or the deer strikes from roadkill? Humans have to clean those up. Nature has actually figured out a way to get rid of that nutrient, the organic matter, but to recycle it back into the system. Because when those maggots have consumed the tissue and then becomes a maggot, then they turn into adults. And those are the flies that you see. Those flies are eaten by other insects. They're also eaten by birds. And those birds then use what they've eaten, right, to feed their young. And so essentially you're recycling dead things through insects and microbes back into the environment and into the ecosystem.
Ah, maggots. It's the circle of life.
Center of the universe. I mean,
the circle of life moves us all, even maggots.
yes.
We have another question. Approximately how many insects, I wonder if you even know this, how many insects have you dealt with throughout your career?
Um,
7. Wait, no.
3. No. Hundreds of thousands?
That's a lot.
I guess.
There are a lot of them. A lot more of us. There are a lot more of them than there are of us.
Tiny fraction. There are people who study a lot more.
What is your biggest discovery to date?
Biggest discovery to date. We haven't published it yet, but I think using microbes to identify how long someone has been dead. That will be a big one. On real humans as part of real death investigations and real autopsies. That will be a big one, I think.
So you were on the cutting edge of using this and although the research behind how it's going to be used.
That's where we're at right now. And the true feasibility of how, if it can be used in death investigations, that's the key. Can you get medical examiners and to actually do this in their lab? Can it become a routine part of autopsies and a routine part of death investigations? We're finding that it's not as easy as one might think.
Or if you're the only guy who can do it. There's only one expert, and it's you. You have to show up at every courtroom.
Every courtroom after it's been vetted, right, scientifically over the years. So yeah, there's been some insect stuff that we've come out with, but I think what we're on right now, and the potential for understanding antemortem, so before death, human health. The way we swab, it just swabs a kind of a random sampling of individuals throughout Detroit. We're getting very different signatures than those that volunteer for health-related studies that you commonly see in the journal articles. And that could be quite provocative.
I cannot wait to see what happens with that. Wow. We have a question from Noah. He wants to know, how did you learn to do what you do now? You were studying insects in Hawaii, and now you're doing this. So how did you make that leap?
It was a lot of trial and error and a lot of work. And it's always a learning process, to continue to learn. And I strive to learn, and I don't give up on things. And so there's some ambition, and there's failure involved, right? A lot of times I've failed. I didn't do something right along the way, from insects in Hawaii to insects on a cadaver to microbes on cadavers now. But you can't be afraid to fail. And the biggest part, and I've been telling my daughters this, is you have to learn from that failure, because you're always going to fail, right? And learning from that failure and not being afraid to move beyond it is probably the key to doing new things that you've never done before, right? And exploring new areas of interest that you don't know a lot about. It just takes some time, energy, and not being afraid to fail at first and learning from those mistakes.
You got to have a lot of confidence and some bravery to just go for it.
That's right. Absolutely.
What inspired you? This is another second grade question. What inspired you to be an entomologist? I know you spoke about going and doing field work with a professor, but was there a moment when you knew, oh, I'm going to be an entomologist?
Yes, that moment was when a professor at the University of Dayton had told me that I would never go to Hawaii and study insects, because I sat in his lab processing samples, looking at insects, washing dishes, and then one day he came in and said, Eric, I might be able to send you to Hawaii to study this insect in the streams. And I said yes.
You had me at Hawaii. So in all of your studies, is what you do very collaborative, or do you find yourself doing it with a larger team or a small team? How do you do that?
It's always collaborative. And now, earlier work, it was more focused on individual effort. But now it's really difficult to do cutting edge science by yourself. And so everything that I've been talking about with the forensics works is completely dependent on many other people where we have a very good collaborative working relationship with different expertise as well. And so not one scientist can do all of this. We brought in 4 or 5 people just for the human microbiome work, just to have that expertise. And I'm only one small part of that.
We're so excited that you're part of it because this is all amazing to know. Ah!
Did it.
That was not 3. I did it. Mrs. Campbell's 5th grade class in Galveston wonders what insect is most beneficial to help the human race.
Beneficial, huh?
Mm-hmm.
Probably the honeybee as a pollinator for food crops.
And also they're cute and they make great Halloween costumes.
They do.
What do you like most about your job?
Being able to do things like this.
You're a communicator. You have to be both.
Absolutely.
I'm sure the students are just about as excited as I am. I don't know, but I'm pretty sure. So what does the future look like for you as far as what you're going to do next? I mean, if there's another larger project that's going in, continuing past work, what's going to happen?
So right now we're working on understanding how microbes affect insect vectors like mosquitoes. And we're starting some new partnerships in Africa to evaluate also how the human microbiome of the nasopharynx affects how people respond to HIV infection or other infectious diseases. So we're going down that route. And then one other one that is it's part of forensics, it's kind of a spin-off, but how insects decompose organic matter is using other insects that say will decompose manure or agricultural food waste or post-consumer food waste. So all of that extra food that people don't eat at restaurants, we have, not just we, but a group of people are studying insects that convert that food waste into insects. And you can then dry those insects and use those for feed for aquaculture, for instance. Aquaculture supplies a huge amount of protein to the global food market. And if we can create a higher and better food source for aquaculture through these insects, while also recycling food waste, so it's not really wasted as much, we're hoping that we can be part of that new science of insects as feed in the future.
So in many ways, you're trying to help save the world, feed the world. There you go.
By understanding, can we use different microbes to make a better insect when it decomposes that post-consumer food waste, right? A bigger, more protein-rich or more fat-rich insect that can be used as food stock, or if you think about the oils, some groups are looking at the oils of these insects for energy sources, much like corn being used for ethanol and fuel and stuff like that. Just some ideas that not just me, but a huge group of people are looking at.
Well, all of those prospects are delicious sounding. Unfortunately, we are all out of time. Eric, thank you so much for coming and speaking with us today. This has been wonderful for you to share your story with us, and I am really excited about the work that you're doing.
Well, thank you, and thank you to all the students that had these awesome questions. I really enjoyed my time today.
All right. Our next event will be on December 21st, featuring STEM role model Dr. Maxwell Rigsby, and we're going to ask Maxwell about what he does as the Chief Product Officer at Gadget Software, at Gadget One. He's an entrepreneur and a technologist. So until then, from Jason Learning, I'm Haley Nelson, and we'll see you next time on Jason Live.